You can tell a lot about a microorganism from an agar plate, besides whether or not it has grown. Colonies formed by each bacterial species will have a characteristic appearance when grown on agar plates. Each of these can be considered separately to help identify the species present. Colony size is important. Some species, such as Serratia marcescens, produce very small colonies, whilst others, such as Bacillus cereus, produce much larger colonies. Fungal colonies tend to spread out and fill the entire plate. Colony shape is also important, which can be considered in different components. These include the overall shape, which may be round, filamentous, rhizoid, or another irregular shape, elevation, which can be flat, convex, or something more complex, and edge shape, which may be rounded, lobate, or filiform. Colonies that are formed by the bacteria that causes anthrax, Bacillus anthracis, have a very filamentous edge. Other colony characteristics to consider include consistency, opacity, and surface type. Staphylococci are described as being creamy, whilst the Neisseria species are sticky. Bacteria with capsules typically form mucoid colonies, whilst unencapsulated bacteria tend to have drier colonies. Staphylococci are usually opaque, while streptococci are usually translucent. Colony colour is also important. Common colours are white, cream, and beige, although colonies can also form in yellow, blue, and green. It's important to know if this colour is contained within the colony or is leached out into the surrounding medium. It can also be useful to know if this colour is water-soluble or not. Some chromogenic agars contain coloured substrates that can aid with species identification, such as Candida chromagar and MRSA chromagar. Another important colony characteristic to consider is smell. Pseudomonas aeruginosa has a sweet grape-like scent, whilst Proteus mirabilis is sometimes described as smelling like chocolate cake. Expert microbiologists will consider each of these characteristics to help them identify the unknown bacterial species present.